Hello everyone, welcome back to my Coursera class, Biochemical Principles of Energy Metabolism. This is the final session of week 5, which is about fat metabolism. So during the previous sessions, we talked about adipocyte tissues, biochemistry of fat molecules, and how those fats, dietary fats or stored fats, can be degraded. And right before this session, we talked about the beta oxidation processes to generate huge amount of ATP molecules from fatty acids. So this session is about fat biosynthesis. Fat biosynthesis. So, fat biosynthesis is quite, quite simple. It's not that complicated. So basically, this is the biochemical conversion of acetyl-CoA, again, C2 unit coenzyme A form can be used for generating fatty acid inside a cytosol. Okay, this is about fatty acid biosynthesis. So interesting point is this one. Carbohydrates like glucose can be used to produce fats in animal cells. Okay, fatty acid degradation product acetyl-CoA can be obviously used for fat biosynthesis. The thing is, even glucose breakdown oxidative processes and the accumulated acetyl-CoA from carbohydrates can be used for fat accumulation. However, there is no reverse pathway from fat to glucose because we don't have such enzymatic systems. So overall, the biochemical formula is like this. The main building block for this fatty acid biosynthesis is acetyl-CoA. Okay, and again, this is a biosynthetic process, which is endergonic process. It needs a lot of free energy, so we have to consume ATP, and this is reductive process. Right? So we need electrons. So NADPH provide electrons. Again, huge amount of the electron or reduced electron donors like NADPH supposed to be used. And palmitate is obviously 16 carbon compound out of 2 C2 acetyl-CoA, total 8 molecules should be used to make one high energy containing palmitic acid. This is kind of the conceptual cartoon showing that carbohydrate digestion inside the mitochondria acetyl-CoA level will be high, or throughout the fatty acid degradation, the beta oxidation processes, so acetyl-CoA level will be high. If the target cells undergo like biosynthetic, like metabolic homeostasis, in that case, acetyl-CoA can be exported out of mitochondria, and in the cytosol, this acetyl-CoA can be used the main substrate to drive fatty acid for energy storage purposes. And those fatty acid out of this acetyl-CoA consumption can be used to store extra energy, in particular inside the adipocyte fat storing cells. And this process is mediated by fatty acid synthase called FAS. Okay, as you can see, this three-dimensional, beautiful, the structural like cartoon. This enzyme is multifunctional and multimeric large molecular weight proteins. Even each monomer is over 270 kilodalton. And as you can see, there are many catalytic sites and activities associated into this complex. So its main job is in the cytosol of adipocyte or the liver. The 16 carbon long chain palmitate can be produced from acetyl coenzyme A as a starting point. So step number 1 of fatty acid biosynthesis is the synthesis of malonyl-CoA. Okay, This is the first step of fatty acid synthesis. Okay, C2, 2-carbon molecule acetyl-CoA. By consuming ATP, we can make this C2-CoA into C3 coenzyme A called malonyl-CoA. Okay, so ultimately this is CO2. Okay, from bicarbonate. So extra carbon can be attached on top of acetyl-CoA, and we have to use a lot of energy. Okay, and this reaction is mediated by acetyl-CoA carboxylase. Acetyl-CoA carboxylase. Okay, extra carboxyl group is attached, and this acetyl-CoA carboxylase indeed is regulated by energy levels, energy condition. So biosynthesis of fatty acid, again, as I said, the first step is malonyl-CoA and maybe acetyl-CoA. Those the substrate can be conjugated, and in the first condensation reaction, the CO2 from malonyl-CoA can be released away. And the second step is reduction. We need more electrons, and dehydration, and one more electrons. Each reduction step, we consume NADPH, electron carriers. So when you see this carbon, this carbon from reduced oxidized CO, and then more reduced hydroxyl, and then CC double bond, and finally fully reduced CH2 hydrocarbon chains can be produced. In this case, those fatty acid carrier is acyl carrier protein, specialized like small polypeptide can be utilized. Okay, basically this condensation, reduction, dehydration, reduction processes are totally opposite compared to beta oxidation processes. So reversal of beta oxidation processes, but this reaction is taking place inside the cytosol, not mitochondria. So fatty acid degradation processes occur in the mitochondria, but biosynthesis takes place in the cytosol. So synthesis and degradation spatially separated. That's the beauty of this tight and the coordinated regulation of fat metabolism inside the cell. So as I said, acetyl-CoA carboxylase is the first enzyme triggering and mediating and catalyzing the whole fatty acid biosynthesis. And this enzyme activity is tightly controlled by energy condition, as I said. In particular, under the energy deficient condition, so like starvation condition, those conditions, ATP levels very, very low. Instead, AMP or ADP level is high. In that case, some specialized enzymes like AMP activated protein kinase, they are activated and then phosphorylate, put extra phosphate on top of acetyl-CoA carboxylase. Acetyl-CoA carboxylase called ACC. In that case, throughout this post-translational modification phosphorylation, ACC becomes inactive. As you can clearly understand, acetyl-CoA carboxylase is the first critical enzyme for driving energy-consuming biosynthetic processes, right? So in that case, under energy deficient condition, specialized kinase like AMP-activated protein kinase stop this reaction by phosphorylating and suppressing the ACC activity. Thus, shutting down the energy-consuming and electron-consuming, highly expensive fat energy, metabolic anabolic, right? In this case, fatty acid biosynthetic processes. This is one of one of the examples showing the tight regulation of bioenergetics in terms of um, um, the energy dependent enzyme regulation. So in this session 5, I gave you in the very beginning, I gave you the sort of overview of adipose tissue, and then the structure of fat molecules, and how fat molecules can be digested from diet, and then how stored fat can be used to supply the free fatty acid. And the core of fatty acid degradation is beta oxidation, and that reaction occurs inside the mitochondria, right? And depending on the energy conditions, fatty acid can be synthesized from acetyl-CoA throughout the ATP consumption and reduction processes, right? And that reaction occurs in the cytoplasm, cytosol. And finally, I gave you one of a clear example showing the tight regulation of bioenergetic processes throughout this post-transplant modification.